For the background of our project, we focus on the idea that plant-based compounds may act as natural alternatives to antibiotics. Because antibiotic resistance is increasing, researchers have been looking more closely at plants since many naturally produce chemicals that protect them from bacteria and pathogens. Our big research question was whether extracts from basil leaves and tomato leaves could inhibit the growth of E. coli, a gram-negative bacterium, and Bacillus subtilis, a gram-positive bacterium. We chose these two bacterial species because they allowed us to compare how plant extracts behaved across two major bacterial classifications. Our group specifically looked at two common plants, Ocimum basilicum, or basil leaves, and Solanum lycopersicum, which are tomato leaves. These plants were chosen because previous research shows they contain biologically active compounds with antimicrobial potential. Basil leaves were chosen because they contain essential oils like lunalol, eugenol, and methylchavicol, which are known to disrupt bacterial membranes. Additionally, tomato leaves were chosen because they contain glycoalkaloids such as tomatine and tomatidine, which can target and inhibit ATP synthase, an enzyme that bacteria rely on to produce energy. Based on what is already known from other scientific studies, we expected basil and tomato leaf extracts to show antimicrobial activity, especially against E. coli. Many previous experiments have demonstrated that basil essential oils can inhibit a variety of bacteria, and tomato leaf glycoalkaloids have been also shown to have antimicrobial, antifungal, and antioxidant properties. Knowing this, our investigation aimed to test whether these plant-based extracts could produce measurable zones of inhibition when compared with our positive control, streptomycin. So overall, the scope of our project was to evaluate whether these natural extracts could serve as potential antimicrobial agents and whether they might contribute to future research on plant-based alternatives to synthetic antibiotics.